Good afternoon. Oh, does this work? Good afternoon. Can you hear me? Maybe not. Okay. Maybe I need the handheld for that. Okay. Can you hear me? Yes. Can you hear me now? Yes. Good afternoon. Welcome to our Focus on Rare Disease Day event on Rare Disease Day, today on February 28th. My name is Greka. I am a physician scientist. I work at the Brigham, Harvard Medical School, and I'm an institute member here at the Broad. And it's a great pleasure to welcome you again to our event. It's been an annual event that's been going on for the last 5 years. It's always very well received, and we're very excited to have all of our guests here again this year. I would like to start with an acknowledgement because sometimes that gets forgotten at the end of a busy event. So I wanted to acknowledge Shannon Humphreys and Amanda Baresi, who have helped organize this event today. So I will just say that at the outset. Thank you very much for your hard work on this. And I think that we are now going to dive right into our event for today. I will act as the MC, so I will introduce all of our different speakers and give a little bit of background, but actually they don't need much introduction because they are all remarkable in their own way, and they have amazing stories to tell us and share with us. So I think this is going to be hopefully an afternoon of great storytelling and community building and thinking collectively about how we can support each other and the community in the rare disease space. So without further ado, I will start by introducing our first speaker. Her name is Patricia Welton. I actually will let her tell her story, but I will tell you my story of how I met Patricia. We actually started this event essentially together 5 years ago, and it started from a very spontaneous phone call. I was walking down the hall at Harvard Medical School and noticed these beautiful portraits of kids, and then read on and realized they were portraits of kids with rare diseases by this exhibit called the Beyond the Diagnosis exhibit. So I went online and looked for more information, found out that it was Patricia who was behind all of this. And I called her because I was giving a talk about rare disease and I wanted to use some of these beautiful pictures. So I wanted permission to use those pictures, and I thought that I should contact her for that. And we had an amazing conversation. Which basically ended up to our agreement to have the portraits come to the Broad and to have them coincide with our first rare disease event that year. That was 5 years ago. And it's been just an amazing collaboration ever since. We've had beautiful portraits, different every year. And of course, as you will hear, they've been, at this point, all over the world. Patricia is the CEO of Beyond the Diagnosis. And it's really an organization that unites art and science to inspire research and innovation of treatments for people living with orphan and neglected diseases. Without further ado, will introduce Patricia Welton, who will give us some remarks. Thank you.
Hi, everyone. Can everyone hear me? That's good. Okay. So, as Anna said, I'm Patricia Welton. We've been coming to the Broad for 5 years now, and I want to thank Anna for that because it's been an incredible experience. We look forward to coming here every year. And this year, we have something really, really special for everyone. We're going to do an unveiling later on in the program. I think you're going to be stunned. Our artist, Hoda Leal, is here, and our patient, I don't know where she is, but Gabby is also here. And so it's going to be wonderful. You'll really enjoy it. So I think I'd like to start with, hi Gabby. I think that what I get asked most often is, why did you think of this? You know, what led you to put art and science together in this way? Because it's really never been done before. So it started because my daughter took 18 years to get a correct diagnosis. And for me, that was far too long. Diagnosis time has always been kind of a pet peeve for me, and we always try to think of ways out of the box that we can engage the medical community in a way that they see our humanity as opposed to listening to a complicated rare disease. So a young lady, a high school student, came to me and she said, I need to paint a portrait for a project, but I want to paint a child with a rare disease. And that set the wheels turning. And I said, well, why don't we start a little exhibit? And we put out a call for art in Rhode Island, and we had 17 portraits. And now we have 120 portraits. We have representing 120 different rare and undiagnosed diseases. We have artists from all over the world and children from all over the world that now participate in the exhibit. First we were at Brown, and then we were at Harvard. After 2 venues, we got a call from CBS Sunday Morning, which I thought was one of my friends pranking me. So they ended up doing a story on the exhibit with 7 million viewers. And I think for the rare disease community as a whole, to get that kind of attention is huge. So I really started focusing on how, we can move beyond what we're doing. And that's what I want to talk about today. Right now on Rare Disease Day, all of those 120 portraits are at a research institute, a medical hospital, or we're at the NIH, we're at the FDA, we're all at a medical school. We're all over the place. We don't have anything left in the gallery. We cannot meet the demand for this exhibit. So, but I want to think bigger. I really want to think bigger, and a lot of this has come because, and some of my friends here and my colleagues know that my daughter had brain surgery 2 months ago. And on Monday, we found out that she has a complication from that brain surgery. She has a left side vocal cord paralysis. So to me, I think about the feelings that I went through during my daughter's surgery and her recovery was still the feelings I felt 5 years ago, and that was of isolation. And it's a huge issue for patients and caregivers. They feel isolated from the rest of the world, and we want to change that. And I think that we've hit upon something here that, we're right now we're living on an island. And 10 years ago, when I first got into this space, there weren't a lot of people outside of the community on the island. And now we're getting, research companies, we're getting research institutes, we're getting biotech, we're getting pharma. And so our island is getting more populated, but we are still an island. And I would very much, and I think we absolutely need the support of the entire world. I mean, this is, to me, if you have 15 million kids in the United States alone suffering without a treatment from a rare disease, that's a health crisis. And I would like to see that as a headline as opposed to this gene therapy costs $2 million for this child to walk. Where are the headlines about what the unmet needs of the rare disease community? There are many. Financial stressors, caregiver burnout. Issues with the schools. If there's something that can go wrong in rare diseases, it's happened to either one of my kids. Both of my kids have Ehlers-Danlos syndrome. So that's what I want to talk about. How we transition from being on our island and taking this exhibit, which I believe is a way to, I'm sure many of you in the rare disease space have talked about rare diseases to someone who has no idea what it is. And what you get in response basically is a glazed over look. If I said to them, my daughter had cancer, there would be an effusion of compassion, and instead, we get more of a perplexed look. It's not that they don't care, it's that they don't understand. What we want to do is I think what we need to do is reach the general public, get off this island. This is a cause. Rare diseases is not a problem, it's a cause. And right now, it's not behaving that way. And that's where we're moving. We're moving towards making this a cause. The art exhibit I have found to be, and I mean, the people who have seen the portraits, everyone who sees it is moved. I get letters that, weekly I am brought to tears. something Anna said during when you filmed the first one, just tears at the way people respond to our children. This is how we need to show the world our children so that the world can respond in turn back to us. If I say to them, hey, my daughter has postural orthostatic tachycardia syndrome, syringomyelia, tethered spinal cord syndrome, and Chiari malformation, people are just going to, what? We really need to bridge that between rare diseases and humanity. And that's what we want to do with the exhibit. We're very, very excited tonight to unveil a portrait. And, I want to thank people like Anna, who recognized right from the beginning that this was important and that this could change things. And and everyone else who has supported us and has been with us for this amazing, magical journey. I want to thank our artists like Hoda Leal, who's here today, and all of our participating artists. I also want to thank our families because they're amazing in allowing us to paint their children and show them to the world because they're making a difference. So that's our goal. That's what we want to do at Beyond the Diagnosis. And we think we can do it. I mean, I think we've exceeded all of our expectations, and now it's time to think big, and that's the next steps for Beyond the Diagnosis. But thank you, everyone, and I hope that you enjoy the unveiling and the rest of the program. Thank you.
Thank you, Patricia.
Was it on?
You can hold on to that, and we will use it later for the, As you may have noticed, the title of our event today is called Putting Patients in the Driver's Seat. So every year we have a little bit of a theme, and I think this is a poignant theme for this year based on the speakers we have attracted and recruited here for you today. And of course, we're starting with two beloved Brodies who have very much been patients in the driver's seat in every sense of the word. They have actually become scientists themselves and have taken on the task of understanding and providing a cure for a rare disease. I'm speaking of Sonia Vallabh and Eric Minikel, who are here with us today. They have a most amazing story to tell, which they have told a few times over in places like The New Yorker and NPR. And I will let them tell you their story. But I call them the prion disease superheroes for the road. They both have now PhDs in biological and biomedical sciences from Harvard University. This is a complete switch, as you will hear. And they trained here at the Broad to do this work. So they were actually also Focus on Rare Disease Day alums, because they actually spoke on the very first event 5 years ago. And they're here back today to tell us all about their story and their progress. So I think we're in for a treat. So without further ado, Eric and Sonia.
Thank you. Thank you so much for having us and for being here. It has been, I guess, a few years since we sort of spoke about our story, and I hope we'll be able to share some updates today on our sort of patient-scientist journey towards preventing a particular rare disease called prion disease. So the first thing to say is that we're a married couple. The story doesn't make sense if you don't know that. Okay. So I'll take you back in time to where this sort of began for us. It was 1 year after we got married in 2010 that we lost my mom very suddenly to a disease that no one could explain. It was sort of resonating with themes we just heard, profoundly isolating. Nobody could tell us what was going on or relate to it at all. All we knew was that she was sort of losing all of her abilities day by day by day before our eyes. And it was only from her autopsy that we learned that she had died of genetic prion disease. And before that information came to us, we had not suspected that this was a genetic disease. So that was a little bit of a shock. And we had to wrestle with the idea that I was at risk and that we had this choice as newlyweds whether to get predictive genetic testing. And we decided to get it. And that's how we got this report in 2011. And it wasn't good news. It was the news that I had inherited this mutation from my mom that causes genetic prion disease. And as we sort of struggled to figure out what that meant, we didn't have scientific training at this time. I was trained as a lawyer. Eric was trained as a transportation engineer. But at a very high level, what we could grasp was that this is, as we saw with my mom, a rapid neurodegenerative disease that strikes at some sort of variable time in adulthood. But once it starts, it's very rapidly fatal within about 6 months to a year. And there was no treatment or intervention or prevention or anything available. The phrase we kept coming across was universally fatal. So that was sort of the state of the art. And since then, a lot has happened. This news catalyzed us to make a huge change in our lives. And we started retraining in biomedical research from the ground up. We took our first jobs in science in 2012. We started our PhDs in 2014. We were super, super lucky to find a scientific home here at the Broad around that time. We've now been here about 5 years. And last year, we defended our PhDs. And we are now sort of running our own independent lab here. And the focus through all that time has really been, how can we tackle this disease? How can we take this very, specific quest and really make progress against it in one lifetime? We've been really fortunate that Broad has bet on us to work on this exact problem and try to get this done. So I think there's still a really long way to go. But in 5 years, I think we've actually come a long way. We now have a therapeutic strategy for this disease that we really believe in. We're betting our life on it. We have a drug development program with a major pharma partner, and it is moving forward towards the clinic, hopefully within the next few years. We'll actually be doing first-in-human clinical trial, and we'll tell you more about that. And we are in sort of ongoing conversations with the FDA about how to do this right. And I think those of you who think about rare disease will appreciate it's not a simple question. There's actually a lot of nuance specific to each rare disease in how to honor the specifics and do the right things. So I'll let Eric tell you a little bit about the disease.
Sure. Thanks, Sonia. So I think that genetic test report that we got at the end of 2011, worst piece of news we ever got, actually contained some good news embedded within it. Because at the point where someone can give you a test report and say, we looked at a single gene, a single mutation, and we know that that means you're going to get the disease, implicit in that is we know the cause of the disease. And so prion disease is a single gene that gives rise to a single protein. And we're also lucky that that protein turns out to be non-essential. So this is a gain-of-function disease, meaning that the protein does do some terrible thing it shouldn't do rather than it fails to do its original job. And in fact, its original job, we don't really know what it is, but it doesn't seem to be that important. So in that sense, we know our target. Our target is to go after prion protein, the one protein that causes this disease. And in the process of going after that target, we've realized we kind of have to redefine what this disease even is. Because when we started in 2012, coming from no science background, beating our head against the wall, trying to understand these jargony scientific papers, what we saw was a lot of terms like Creutzfeldt-Jakob disease, Gerstmann-Straussler-Scheinker disease, these sort of alphabet soup of things that kind of make people want to stop listening, including us at the time. And what we've realized is these terms mean little to nothing. They are just historical accidents that some doctor discovered this, it got named after them. The true name of this disease is prion disease. It should be named after the gene or the protein that causes it. And so our disease is unique in a few ways. So we have these different, quotes, clinical subtypes with these different historical names. There's also different ways you can get the disease. So not all cases are genetic. Some are just happen spontaneously or called sporadic. It can also be acquired very rarely. It can present in a lot of different ways. And what we've come to realize is if you boil it down to the genetics, the molecular cause of the disease, this is one thing. This is just prion disease. And we have our target. And so basically, our goal is to go anywhere upstream of where things start going wrong with this protein. So past drug discovery efforts had aimed at misfolded prions or how do they kill neurons. And those turned out to be difficult targets for a lot of reasons. But if we aim upstream, then we've got a target that should be the same across anybody who's at risk for this disease. And it's a target where we know the sequence or we know the structure, and we have ways to measure it and go after it. So there's excellent evidence from years of studies in animal models that if we can lower the amount of this one protein, that should delay the disease. So animals that produce more of the protein get sick faster, less of it, they get sick slower. And if they have none of it, they're totally invincible against this disease. And meanwhile, people have also made knockout animals. So there's knockout mice, I think 7 different lines of them. There's knockout cows, knockout goats. Using these large human genetic data sets at Broad, we've been able to identify, I think at last count, 8 different humans who are deleted for 1 out of 2 alleles of this gene. So we've never yet seen a human who's a total knockout for prion protein. But as far as we can tell, having at least less, and from the animal models, probably none of this protein is actually fine. So the question is, how can we do it? So what is the strategy that is realistically going to get there in our lifetimes that can lower this protein? And I'll give you a hint. It's not necessarily something novel, right? You go to funding agencies, they always want to say, give us novel mechanism, give us insights into disease biology. And actually, a lot of that stuff either doesn't pan out, or if it does, it's long after you're dead, right? What we have today is we know the causal gene here. And if we can find a modality, a type of drug that's already been tested in the human brain and been shown to work at this kind of problem, then we're going to be many years ahead of the game compared to if we had to start with something novel. And lucky for us, there exists such a modality. So antisense oligonucleotides, short strings of chemically modified DNA that are programmed to have a sequence that binds the RNA of your gene of interest, can lower the amount of one target RNA and thus lower the amount of protein produced from that gene. Back in 2014, when we were kind of kicking off this project, ASOs had just started to have success in the human central nervous system. So it had been figured out that you can dose them by lumbar puncture into cerebrospinal fluid. Lumbar puncture, by the way, not as scary as it sounds. We've both undergone them for research. Sonia will tell you more about that in a little bit. And they distribute across the brain, and they're stable for weeks to months. And it is actually a realistic way of chronically dosing a drug to treat a disease. So since then, since we launched this project, I think our suspicion that this was a promising modality has been validated by the approval of nusinersen for spinal muscular atrophy and by successful early phase trial results for Huntington and SOD1 ALS. And when we were first launching this project, I think one question that we got from people was, well, if this therapeutic hypothesis makes so much sense, and your target makes so much sense, and this modality exists, won't somebody just do it, right? Why isn't industry all over this? And maybe the answer is different in a common disease. Maybe in Alzheimer's disease, the rewards are so great that even things with like a really low chance of success, you can afford to roll the dice. But at least what we've learned in this rare disease is that we really need to hand everything over on a platter, right? We need to do a lot of groundwork that industry is absolutely not going to do. And in many cases, NIH isn't going to fund either. This is stuff that we had to fundraise from patients and families in our network and the generosity of the Broad Institute to make our rare disease what industry would call developable, to say we've got a validated target, we've got a patient population we can go into, animal models we can use, a biomarker we can measure in these people, we know how to screen for people who are at genetic risk, recruitment tools, things like that. So the last 5 years have been basically us trying to lay that groundwork. And I'll just show you 2 slides of just a little bit of the data. So this was the first mouse study we ever did. These mice were at the Broad Institute. We had finally convinced Ionis Pharmaceuticals to develop ASOs as tool compounds against the mouse prion protein gene and send them to us here. And in a mad scramble to get these studies off the ground, the day 0 on this plot is actually the 4th day of our daughter's life. So there was a scramble to get these studies done, and we weren't willing to delay them at any cost. We basically infected mice with prions, gave them 2 doses of ASO, and the ones that got the active compound lived 60 to 80% longer. It was an incredible result. And this corresponded to an extension of healthy life. We had actually pushed off the accumulation of symptoms of the disease and weight loss and so on. But that's not the end of the story. And one reason why that's not the end of the story is that there was a hitch, which is that when you treat has a lot to do with how much good and what kind of good you can do. So we've repeated this result in, I think, over 1,000 animals now, varying the dose and the strain or type of prion disease and so on. One of the variables that we found matters a lot is how long do you let the disease progress before you intervene with the drug. And if you intervene early with a chronic treatment to keep PRP low, we can triple the survival of those animals. So that's the curve at the far right. But if we wait until the animals have really obvious neuropathology in their brains and are at the cusp of actually coming down with rapidly progressive symptoms, the amount of good we can do is much less. So in this study where we're following them to a symptomatic endpoint, we only were able to delay the accumulation of symptoms by about 20%. And if you let them go until terminal disease, you can push that off a little bit longer, but you still cannot do nearly as much good as you can if you intervene early. So this brings us back to the human question, right? So these are mice, but in humans, we have patients who've just been diagnosed who are sick, and we have patients who might know 20 years in advance that they have a high-risk mutation. What can we do to give ourselves the best shot on goal of showing that this drug actually works?
So I think this has actually been one of the more interesting and surprising parts of our quest is we came in on day 1 saying we need to find the molecule that's going to treat prion disease. And we absolutely do need to do that. But that turns out to be just this one piece of this larger puzzle. And I think you could find the molecule and still not end up doing the right things with it if you're not careful. So Eric just talked about when you treat being really important. I think one vivid illustration of that is just reflecting back on what we saw in my mom's disease course, which was less than a year long. And even to say that it was 10 months, I think overstates the case because she lost her abilities so fast after first symptoms, I would say over the course of about 4 to 6 weeks, and was on life support by summer. So when I look at this timeline, there's no point here where it was really clear that we were up against a serious neurological disease, but there was still meaningful quality of life there left to preserve. It all just went by in a blink. And that to me is scary because I feel like if it was me, there's sort of no point on this timeline where I would say, okay, if a drug exists to stabilize me in this condition indefinitely, sure, I want to take it. That's actually a longer disease course than average in prion disease. 10 months is longer than average. Most people don't live half a year from their first symptom. And if you look at the few clinical trials that have been done in this disease, you can really see those challenges play out. People are recruited to these trials at a point where they are already on life support measures. And to try to do meaningful good for people at that point is so hard. As Eric said, I think we're looking at sort of, thanks to genetics, right? Thanks to predictive genetics, we're looking at 2 different patient populations. Folks who we actually can't identify until they're sick, and people who have genetic mutations that they might be aware of long in advance. And I think that really is an opportunity that we have to take seriously. So as we think about what would it mean to leverage predictive genetics into trying to treat earlier and keep healthy people healthy, one thing that we have encountered, a model that we've encountered for pre-symptomatic trials, is this idea that you go out, you recruit people based on having the mutation, you randomize them to drug or placebo, and you just follow them till they get sick. I don't love that trial design personally. Even if I did, we've crunched the numbers on this, and prion disease is sufficiently rare and has a sufficiently variable age of onset that it actually is infeasible to do it that way. We actually couldn't get an answer out of that kind of trial, even if there was a sponsor out there who was willing to follow people for 20 or 30 years to get drug approval for a rare disease drug. So I think that's a non-starter. Where does that leave us? If we can't treat healthy people and follow them to a so-called clinical endpoint, follow them until they're sick, then we need an alternative. And I think that alternative ends up being a so-called biomarker, a molecular measurement that we can trust that tells us if the drug is doing its job or not, sort of by proxy. So in 2017, we launched a clinical study at Mass General Hospital with the goal of bringing in genetic prion disease mutation carriers and controls and studying the potential biomarkers in this population. It was really gratifying to see our community rally around this study. We announced it on Facebook, and 2 days later, more people had volunteered than we had the money to see. So we've been slowly trying to expand and keep people coming back. I think it's exciting to be building a little team. So I'll quickly summarize at a high level what we've learned from analyzing the samples that these folks have really generously donated to us. I think based on some of the data that are out there for diseases like Alzheimer's and Huntington's disease, a lot of people had assumed before we ever did the study that carriers are in a sort of ambiguous limbo state. Even if they're not sick yet, surely there are things going wrong. Surely if you take their blood or you take their spinal fluid and you subject it to the right analysis, you'll see that there's signs of disease. Like at some low level, there's signs that neurons are dying or that something else is going wrong. That's not what we see. And this has been interesting. At the stage where prion disease is manifest, where patients are symptomatic, markers of things like neuronal damage go way up. But when we compare our healthy carriers to matched controls, they all look normal. On basically every front that we can measure, they look normal. Another thing that we've been looking at is prion protein levels in spinal fluid. So Eric made the case to you that prion protein is at the heart of this disease. The genetics are super clear. It's a smoking gun. And we just want to reduce the amount of prion protein in the brain. If we need a marker to show that we did that, could that marker be prion protein in cerebrospinal fluid? Well, in order to see whether this could function as a biomarker of drug activity, we asked people to come back 2 to 4 months apart, get a lumbar puncture once, come back 2 to 4 months later, get another lumbar puncture. They've been incredibly generous, and they have done that. And what you're seeing here is prion protein levels in one person over that time period. And we got really lucky here because this was a bit of an unknown. Like, would we be able to measure levels of this protein reliably? Would the levels be stable in one person over time? Because if you want to show that a drug made a difference, you need a stable baseline. Would the story be the same across these different mutations that cause prion disease? And basically what we found is yes, yes, and yes. This seems like a biomarker that's behaving. And if we could lower the amount of this protein with a drug, I think we would see that. And we've now, thanks to our amazing cohort, been able to follow people even longer. They're willing to come back a year later and give spinal fluid again. And we continue to see that the levels of this protein are really nice and steady. And if we could hit them with a drug, I think we could convince ourselves that we'd had the effect that we wanted to have. So high level, and this study is ongoing, and I hope it will, we'll continue to broaden the cohort and follow people longer as well. But what we can say today is that carriers appear healthy from everything we can measure, which I think gives us this awesome opportunity to keep people healthy if we can intervene early enough. And this is kind of not where I expected our quest to take us, but this turns out to be a project in sort of multi-sector coalition building. I think treating healthy people before disease strikes turns out to be a pretty unusual and a pretty radical idea. And everybody from the carriers themselves to pharma and biotech partners, to doctors, to insurers, to the FDA, kind of has to be convinced and brought along if we're going to be able to make this a reality. I'll say a little bit about our conversations so far with FDA. So in 2017, we talked to them for the first time about this pretty simple idea of reducing the RNA that encodes prion protein with an ASO drug like Eric showed you a cartoon of, and then just measuring prion protein in spinal fluid, and if it goes down, could that alone be the basis of provisional approval to get a drug to healthy people to try to keep them healthy? It was a very nerve-wracking conversation. I think this was probably the scariest meeting we'd ever been to. And we were both really impressed by what a warm reception this idea got. I think there's kind of a lot of momentum right now in the idea of preventing brain disease, moving further upstream, trying to save the brain because it's very hard to rebuild it once the damage is sort of already on its way. But this is a pretty tall order. You need to have the right tools. You need to know exactly which molecule you need to be hitting. And we made the case to FDA that in this disease, those stars have aligned. And they basically said, you have your work cut out for you. You have a ton of experiments you need to do, and we're going to look at the data together. But we'll advise you on how to try to get this done, because preventing this disease makes sense. So where does that leave us? We're working with Ionis to try to move prion protein lowering ASO towards the clinic as efficiently as we possibly can. After those 5-ish years of assembling the whole platter of different kinds of data and different kinds of tools to convince them that this could be done and to de-risk the target and the project, we convinced them to sign on and to commit to developing a human version of the drug that we can take to the clinic. So that has been a huge milestone, and they've been great partners, and there's still a ton to do, but it's exciting to see the momentum build behind this. Yeah, this is that platter, I guess, just like a callback to all the different things that were on our homework list the first time we ever met with them. And we left that meeting thinking, we don't know how to do any of these things. But I think as a function of having to do them, we learned. And I think that's what's emerged from these past several years of work is realizing that if we want to move all the way from figuring out what is the molecule to having a human clinical trial structured in a way that makes sense and launching, we need to be totally vertically integrated. We can't afford to say, oh, but we don't know how to do mouse studies. We just have to figure it out. I think another thing that we're starting to put some thought and bandwidth into is what's going to be next. ASOs can make a tremendous difference. They can lower PRP by, we think, about 50%, and they can, as Eric showed you, maybe triple the life of a prion-infected mouse. That's awesome. All of those mice still die of prion disease eventually. But if we could lower the protein levels even more, I think we could buy them more time. And I think there comes a point where you've extended life long enough that it's a functional cure. So are there other tools out there? Maybe not, definitely not as close at hand, but on the horizon that could be drug number 2 in our lifetime. This is something we think about increasingly these days. And I think I've been impressed at the way we've been accused of putting all our eggs in one basket with the ASO program, but I stand by that decision 100% because by assembling all of the things on that checklist, I think we have sort of built the ramp to conversations about drug number 2 and sort of shown that the therapeutic hypothesis works and we've got the pieces in place to advance it. And I hope that that will just help us move faster on the next projects. And then finally, sort of like an ongoing, ever-present project of ours is to engage the patient community. A lot of people have been told not to get predictive genetic testing, and I was told that too. And I understand why. But I think there's a whole spectrum of preference out there, and we need to honor that whole spectrum. And there are some people who might never want the test, even if the drug is approved and available and sitting on the shelf. And there are some people like me who, no matter what you tell them, they want the test. And even if there's absolutely nothing you can do about this universally fatal disease, they want the test. And then there's a lot of people in the middle to whom I think it is meaningful that we are living in an age where research is not just for the next generation, where what we can do is changing, right, year to year, and where targeted therapeutics for rare diseases are a reality, and they're coming out of this city every year. I think I understand why there's an urge, a desire to be very cautious about raising false hope, but I think it is sort of like equally irresponsible to obscure real hope. So there's a delicate line to walk there that we are constantly trying to honor. A few closing reflections. It's been interesting being patient scientists. We don't fit well anywhere. I think if there's one place we do fit, it's Broad, and we're super lucky to be here. But we sort of feel every day the way that academic research incentives and patient incentives are misaligned. And that's a tension that's probably never going to go away, and that we just have to learn to manage. But I think that it's been worth it because if we weren't in this race, I think no one would be talking about preventing disease. And success would still be defined the way it was when we got into this field, where people were saying, if we treat symptomatic patients who have lost all of their quality of life, and they spend an extra 2 months alive in a hospital, that's a successful clinical trial. So I think getting not just our voice, but those of the other patients with whom we've been able to build a network into the conversation has actually completely changed the goal of drug development. And I, even though I think we have a very hard job, I think that makes it worth it. These days we hear from folks who are also crusading against a rare disease in various forms. And I think there's a lot that we share in spirit, including this sense that, actionability isn't just defined by the drugs that exist today, that there's actually a lot we can do. And that especially in a rare disease, right, every person who feels motivated to get involved makes a huge proportional impact. I think it's interesting looking across the spectrum. For us, there was this particular way of getting involved that we thought made sense because the basic biology of prion disease was understood. A certain amount of infrastructure already existed. There was already a patient support group. There was already a biobank. And what we felt was missing was the link between understanding the disease biology and saying, hey, we could leverage that and make a drug. So those are the people we choose to try to be. I think every disease has a slightly different landscape. It's in a different state, as of today, with different resources and different needs, and the biology is asking you for something different. So it's like, I think, a really interesting balance of the things we share and the ways that all of these things differ in their details, and trying to figure out how to honor all of that complexity and sort of move forward together. It's been an interesting few years, and I hope that we'll have more updates to share in the future. Thank you so much.
Eric and Sonia, thank you so much. This was amazing, and you can see the incredible progress that has been made in the last 5 years since they last shared their story with us. And I hope you can see when we said patients in the driver's seat, we're talking about patient scientists in this case, in this remarkable journey that Eric and Sonia are on, and so many other patients are on. And so that sort of brings us to the final segment of our Focus on Rare Disease Day today, and that's our keynote lecture. We are very lucky to have Tania Simoncelli here from the Zuckerberg Initiative to speak to us about Rare as One, an amazing project that they have launched to strengthen patient communities and accelerate research across all different rare diseases. So Tania is now the policy director for CZI Science and the lead for the Rare as One project. But she has a long history, some of which is very much linked to the Broad. For 20 years, she's really designed advocacy strategies and policy solutions across the intersection of science, technology, law, and ethics. She had previous roles here at the Broad, actually, recently. She was part of the White House Office for Science and Technology Policy under President Obama. She had roles at the FDA as well as the ACLU, and in fact, she's quite well known, named by Nature as one of the 10 people who mattered in 2013 for her work in spearheading the ACLU's successful Supreme Court case challenging the fact that human genes can be patented, which was really a remarkable case. So Tania has had this incredible trajectory as an advocate for patients and science and policy across many different areas. And it's really an incredible treat to have her here today to share with us the inspiration and the early successes of this amazing project called Rare as One. I just wanted to say that, as we sit here at the Broad, we are part of a long legacy and a long history of, it took us, more than a decade to sequence one human genome. Now, in our facilities here at the Broad, we can sequence a human genome about every 8 to 10 minutes. It's a remarkable progress, and we are able to help a lot of people by, sequencing the genes and finding the genetic underpinnings of several diseases, many of them rare diseases. So now we are called upon to do more. Beyond the diagnosis, as Patty says, we have to use our resources to really be able to build the kinds of projects and coalitions that will allow all patients everywhere to benefit. Of course, these rare diseases affect children, many children as well. So patients to benefit all around the world from understanding what these errors in the genes mean, what are the mechanisms that are disrupted, and how we can actually build therapies. And to do this by building communities, scientific and patient communities, who work together at scale to be able to implement this strategy. I think it is Priscilla Chan who said, we have the ability to cure human diseases in our lifetime. It's a lofty goal, but it's something that is achievable if we actually set our minds and hearts to it. And I think that's really what we are faced with. That's what the broad community can come behind. And I think that's what our friends at CZI are also working on. So without further ado, I will introduce Tania. But I will say that she will show us a video for a few minutes first before she makes her remarks. So I just wanted to foreshadow that. So yeah, Tania Simoncelli, and Rare is One.
The life that you thought you were going to have is going to probably be significantly different.
If you're called to do this, you have to accept that your child may not benefit from the work that you do.
I had to let it literally go through my body and say all the nevers. You know, Amara will never have a career. She will never play soccer. She will never be able to tell me she loves me.
If I could halt the progression of my disease and stay right here forever, I would not utter one complaint ever in my life.
It took almost dying in medical school to realize that I couldn't just hope that the current system would work. I said, I'm going to dedicate the rest of my life, however long that may be, to trying to cure this disease.
Every seizure feels like it's a lifetime. We're veterans at this point. In 25 years, we've seen over 40,000 seizures just in this kid.
I have about 31% lung function. I spend about 4 hours a day on medical treatment. And there really wasn't anything in the pipeline that was poised to help people like me. That was just a reality that my family and friends and I could not swallow.
I started having really severe multi-organ failure where my family actually called in a priest to administer my last rites.
I might go deaf and blind at some point. Oh my God, like somebody is just gonna turn off the light.
We're at the cutting edge of science. The patient community is pushing the boundaries. I met two mothers, one in New York, one in Switzerland. We joined together, we started the foundation. So that helps us understand our disease and it helps us build therapies that could potentially save patients' lives.
It was my son who had the idea. Why don't we start selling cupcakes to raise money to cure Cal?
I had all this data from my tumor. It was sitting on a hard drive. And I thought, what if I share it? I start thinking, okay, what's next? Now I work directly with patients and communities to access their medical data, so they are holding the key.
You know, as a patient, you don't realize the assets that you can bring to a situation. We sent out an email, and within that first week, we ended up raising over $40,000. We now have 12 active research projects to help make it so that the only thing that stands between us and breakthroughs and cures is science.
I just wanted to be a part of the group of people that were trying to find the answers. And so I went back to school and ultimately got a PhD. We found a drug on the market, and within 2 weeks had a 95% reduction in her seizures. And at the end of the day, honestly, my money's on the patients.
And now it's been over 5 years that I've been in a remission on this drug that I identified through research. It was right there at my neighborhood pharmacy that I'd always passed by, and no one had ever thought to try it, but it's saving my life.
I called Cal's doctors, and she said to me, I know what you should do with the cupcake money. You need to help the families get to the gene therapy trial. And that was what we started to do. We're not going to be able to save Cal, but we're going to have a front row seat to the biggest medical breakthrough in a generation.
I'm Onno, I have NF2, and I'm one of the 7,000.
My name is Nasha, this is Amara, and we are one of the 7,000.
And we are one of the 7,000.
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 I'm one of the 7,000.
We are one of the 7,000.
I'm one of the 7,000.
Great, thank you. Thank you so much. Thank you, Anna. What a wonderful event. I'm so happy to be here, back at the Broad. It's a place that feels like home to me, especially with Sonia and Eric, who I just am always just so inspired. You guys really should be the keynote, not me, because this is about patients driving progress, and you guys are doing something that is truly astonishing. And I just think it's really just such an amazing example of what patients can do. And that's what really inspires me every day, and is what has inspired this program, the Rare as One program. And I thought I'd step back a little bit and tell you a little bit about really just how this came to be and what we're trying to do. So the Chan-Zuckerberg initiative, oh, and I should also give a shout out to Joanna Rudnick, who made that beautiful video. And to sort of pile on to Patricia's comments about the importance of art and film and storytelling, bringing humanity to this space. And it's just so incredibly important because, you know, we live in a very complex world. Most people don't know that rare is common, that so many people are affected by these diseases and are grappling with this every day. And I think Joanna really did a beautiful job. She's a really wonderful documentary filmmaker and good friend. So big shout out to her on that. CZI, to back up for a second, is a philanthropic organization. It was founded by Mark Zuckerberg and Priscilla Chan 4 years ago. So we're still very, very new. And they founded it following the birth of their first child, Max. And it was really born out of this conviction that Mark and Priscilla had that a better world is possible for their daughter's generation. They wanted to create a more inclusive future, a future for everyone. And sort of foundational to sort of all the work that we do, whether it's in our science division, we have sort of 3 pillars that we work in, whether it's in our science division where we're working to cure, manage, or prevent all disease by the end of the century, whether it's in education where we work to promote whole child learning and education, or whether it's in justice and opportunity where we work to make sure that that the experience of justice and opportunity is an experience that's for everybody and not just the lucky few. We have this sort of idea that Priscilla often says this, that luck is not a national strategy, that we need to build strategies that really take luck out of the equation for every person. And what she means by that is we need to address the real problems, the deep systemic challenges that are confronting these complex societal problems that we're working on. And that's sort of how we think about what we tackle and what we take on, where we take on long-term problems. We take on, we don't shy away for the really complex issue, the really thorny challenges. And we also work in a really unusual way. CZI is unusual in comparison to other philanthropic organizations in that most philanthropies focus primarily on grant making. But we really see ourselves as working at the intersection of philanthropy and technology and advocacy. And we believe that the solutions to problems lie with the communities who are most impacted by those problems. And so we look for bold ideas in those communities, and then we look to see how we can leverage our combination of capacities, not just in grant making, but also in technology and engineering, in policy and advocacy, to support those solutions and to replicate and scale those solutions to drive meaningful change. So CZ Science, as I already mentioned, has this really audacious goal of supporting the science and technology that will make it possible to cure, manage, or prevent all disease by the end of the century. That's 80 years. That's not that many years, but if you think about where we were 80 years ago, it starts to put things into perspective. Like, we didn't even have, antibiotics were not even really on the really available 80 years ago. So look how far we've come. But when I got to CZI 2.5 years ago, I embarked on this sort of inquiry and to try to answer this question, which was, how can we support and leverage the power of patients to accelerate research and drive progress against disease? I felt like if we were really going to make good on our mission, that we couldn't do it without not just partnering with patients, but really understanding the full potential of patients, those communities that we are ultimately aiming to serve, the full potential of patients to really accelerate research. And so when we started to look in this space, what we learned, and we talked to dozens and dozens of patient advocates, not just rare, but ended up focusing more and more on rare as we went. And what we noticed is that more and more patient communities are playing an increasingly active roles in accelerating research. Some of them doing the things like, very few, but some, like Sonia and Eric, changing their careers and actually becoming scientists. But the point is there are a lot of ways that patients are actually doing more and more, and not just as partners in research or participants in research, say in research studies, but really as drivers of research. They are doing things like you saw in that video. They are raising funds. They're shaping research agendas. They're bringing together researchers. They're actually soliciting researchers and recruiting them to their diseases. They're making it possible for diseases to be studied that couldn't be studied by building research-enabling infrastructure, things like registries, patient-led registries and biobanks, developing cell lines in partnership with laboratories. They're then going about, they're doing what Eric said so eloquently in the rare disease space. It's becoming clearer and clearer to patient communities that they have to do this. They have to deliver it on that silver platter. They have to build the research-enabling infrastructure, make it possible to actually study their disease. Because if you think about it, in the traditional academic research model, the incentives just aren't aligned. If you're going to study, let's say, an ultra-rare disease, you have to find the patients around the world with the disease. You have to collect their specimens. You have to, bring them together as a community. There's a whole bunch of work that has to be done. But if the patients can do that, it can really, really accelerate progress. The challenge is that this is sort of this emerging model, right? This kind of, I presented it as sort of a pyramid. I used to have it as kind of a continuum. It's sort of a continuum, but these are sort of some of the key stages that I think are somewhat generalizable that patient communities are following. They have to have a strong patient community. They then work to sort of reach out to researchers, build research networks, identify researchers who will tackle their disease. They look to build, researchers registries and biobanks. They look at ways to engage industry and help drive clinical trials forward. But the problem is that there are huge barriers and challenges to patient communities doing this kind of work at each of these stages. And so the Rare as One project is really focused on how can we take our collective capacities that I just described in funding or grant making and advocacy and engagement and in science and technology, how can we actually make this process easier? What about these things are somewhat generalizable? How can we build capacity into patient communities? How can we create roadmaps? How can we support them in this work and help lower the barriers to entry? And so when we thought about this, we thought, well, where do we start? And we got really interested in this kind of space of patient-led research networks and sort of this really important place where it seemed to us, the more we talked to communities, we learned that one of the problems that communities had is they were really good at building their patient community and recruiting people into their space, and then they were actually really good at raising funds, but then they didn't quite know where to put the funds. They didn't know which researcher to give the funds to. And we were really inspired by the story of David Fajgenbaum as we started to think about how could we help patients do that work better, actually come up with really strategic ways of directing research dollars and actually bringing the research community together. And if you're not familiar with David Fajgenbaum's story, I encourage you to read his book that he just put out a few months ago, Chasing My Cure. It's a really wonderful story. But in a nutshell, in 2010, David was in his third year of medical school at the University of Pennsylvania, and he woke up one day with flu-like symptoms, and he ended up in the ICU for, I think, 7 weeks, and his organs started shutting down. And luckily for him, luck in the equation again, luckily he was a third-year medical student, there's that, but he was also at the University of Pennsylvania, and he was diagnosed actually fairly quickly compared to most Castleman disease patients with idiopathic multicentric Castleman disease. And he was given chemotherapy, and he went into remission, but only for a short time. And he ended up having 4 relapses with this disease, and each time bringing him very close, very much to the brink of death. And in between his relapses, he spent his time learning all that he could about the disease, and he was really dismayed when he found out that there were actually a fair number of researchers working on this disease. It had been identified in the 1950s. But they were doing so in isolation. There was no coordinated anything. Like, most of the researchers had never talked to one another. There had never been an international research meeting on this disease. There was no one aligning researchers or trying to develop any sort of agreed to priorities on like which research mattered. And what he realized was there was no one with the interest of like, with the sort of goal of solving the disease. And he's like, whose job is it to solve the disease? And so he actually called every single researcher that he could on the phone, and he wrote to them all, and he asked them, would you join? First he asked them, why are you doing what you're doing? And like, how is that gonna help us understand this disease? How is it gonna help us solve the disease? And some of them were doing really important and really interesting work, but there was no coordinated strategy. And he realized that's what I wanna do for the rest of my life, whatever time I have left. I am gonna go out and I'm gonna organize the research community. And so he asked them all to join a discussion board. It's like very low-tech version of a discussion board. And he asked them really simple questions. What do you think are the top 3 research priorities for this disease area? Not for what you want to work on, but for the field. And what research is being done and what isn't, and what are the priorities? And he ended up pulling together an international research meeting, the first ever for Castleman disease. And the results were astounding. In just bringing people together to talk and to actually start to collaborate in a very short time, what happened was what was emerging through these discussions was that the whole model for the disease was really wrong. And the research community started to realize through these discussions that they had it sort of upside down, that it didn't really make sense to be treating this, at least for some patients, fundamentally almost like as a cancer, that really this disease was fundamentally a disease of a hyperactivated immune system. And they started to better understand the underlying biology by developing a research strategy that really looked at figuring that out. Well, the impact of this was really important for David's life, because somewhere around that time, as these conversations were getting going, and these sort of realizations were happening collectively across this community, he experienced his 4th relapse. He didn't want to take any more chemotherapy. He actually wasn't sure he could survive any more chemotherapy. It was just too toxic at that point for his body. So he convinced his doctor to prescribe an immunosuppressant, that sirolimus, a drug that had never been used in Castleman disease, but has been commonly used for people undergoing kidney transplants. And he recovered, and he's been in remission ever since. And it's been more than 6 years, and he had 4 relapses of this disease within 3.5 years. And this drug seems to be working on other Castleman disease patients, and not all of them, which is an indication that there's something else going on. There are subtypes of this disease, at least 3 of them. They don't actually know how many. There's been an extraordinary amount of research now coming out of this work that David's doing. He's now brought together more than 1,000 patients, researchers, and clinicians that are all working together. They have developed a unified, prioritized research agenda for Castleman disease, and it is completely transforming the outlook of this disease for patients. And sirolimus is actually, there's now an active clinical trial trying to investigate the efficacy, whether that drug actually works on other patients and which ones. So that was a really inspiring story. It's just one of many, but this one was really inspiring to me because I realized that what was so important about what David did is he recognized that he had to build the community. Another thing interesting about David is people say to me, well, but he was an MD. Well, he wasn't really an MD yet. He was in medical school. So he had this sort of extra insight. But what was so interesting is like he actually didn't go on to do his residency. When he finished medical school, he decided to go to business school, because he realized this isn't really a scientific problem per se. This is a problem, a social problem. This is a business model problem. This is like, so he actually wanted to study it as from the perspective of business. So we launched the Rare as One project in June of last year. And as part of our launch, we invited rare disease organizations to apply to join the Rare as One network. We were really thrilled. The whole idea behind the funding was we were inviting them to apply for funding, not just funding, but we were also going to give them capacity building support to build and sustain collaborative research, patient-led collaborative research networks in their rare disease area. We received 287 applications from 43 states in the first round of applications. Most of the organizations were less than 5 years old. Most of them were led by patients or parents or family members of patients. Most of them were doing this work completely unpaid, often with full-time jobs, and often having to take care of loved ones with rare diseases. And the majority of these organizations had budgets of less than $300,000. We invited 83 of these groups to come to submit a full application. And then earlier this month, we announced the awardees. We actually initially planned to fund only 10 groups, but we were so blown away by the quality of the applications and in particular, the quality of the patient leaders leading these efforts, and that we ended up deciding to fund 30 groups. So we actually tripled the program. And these are the groups. That's a lot to take in. They're all described on our website. We have a little blurb on each organization. I'll give you a little bit of a sense. Each of these groups is going to receive $450,000 over 2 years to build and strengthen collaborative research networks in their area. They're being asked to host at least one international scientific meeting with the goal of developing a unified research agenda in their disease area. The leaders have an incredible range of backgrounds. One of the most inspiring things to me in reading these applications were the ways that the patient leaders understood fundamentally that they didn't need to be a scientist to do this, that it was a problem of organization, that they could leverage and apply whatever backgrounds they had. And there were a couple that had some kind of health-related background or healthcare-related background. We have a surgeon in the mix who's one of the leaders. But we also have an actor. We have people with communications backgrounds. We have people who are former executives, entrepreneurs. We have a former professional basketball player. And a lot of stay-at-home parents who understand that they have unique skills, and a lot of what their value is really deeply understanding the disease that they are dealing with every day, day in and day out. So I can't go through all 30. I'll just give you a couple of examples. The Cure Grin organization, this is an organization that's being co-led by 2 parents with 2 children with grin disorders. Grin disorders affect one of 7 different grin genes that are involved in learning and memory. So these children have developmental delays. Some of them have very severe intellectual and physical disabilities. 500 people have been diagnosed worldwide with grin disorders, but we expect there are probably many more children with these that just remain undiagnosed. Denise, Denise is there with her child. She has a marketing and communications background. She's worked for a number of global companies like IBM and Cisco. And Keith, the other parent, is an entrepreneur and author, and was a former VP of a company. So to give you just a sense of sort of where they're coming from, HPS, the Hermansky-Pudlak syndrome network, this is one of the groups that actually has been around the longest in our cohort. HPS is a single gene disorder that involves albinism and legal blindness and a platelet defect that causes bleeding and in some cases fatal pulmonary disease. And the network was founded by Donna Apple, that's Donna with her daughter, back in the 1990s after her daughter hemorrhaged and sustained a traumatic brain injury from acute blood loss when she was just 2 years old. And what is so just astounding about what Donna has done is that at the time that her daughter was diagnosed, she was told no one knew of anybody else with this disease. She could not find any patients. This was before the internet. This was before, the kinds of things that we have now where it's a little bit easier. And since then, she has now connected 1,300 patients from around the world. And one of the things that's so inspiring is she's doing extensive outreach in Puerto Rico, where there's a higher incidence of this disease due to a genetic founder effect. And she's doing things like organizing school buses and vans to pick up affected individuals to bring them to conferences. She spent an enormous amount of time tracking down patients that had been forced to move after Hurricane Maria. So she's just an incredible mobilizer and community builder, and we were just so inspired and thrilled to have her as part of the network. Tango 2, this is a disease that presents as muscle breakdown and metabolic crises and life-threatening cardiac arrhythmias, and it's usually brought on by illness. And this foundation was founded by these parents, Mike and Kasha Morris, in 2017 after their son was diagnosed, so relatively new organization, just a couple of years old, but in less than 2 years, they have engaged the entire known community, affected community with this disease, 70 families from 16 countries. They've established a registry, they've catalyzed research, they've helped fund a natural history study, organized a first conference, et cetera, et cetera. I think I'll stop there. But we also understand, we can't just give these groups funding. That's not sufficient to make these groups sustainable. These are tiny organizations, patient-driven organizations. So we are building out a capacity building program. And what's important to know about this is we're looking to learn from the groups as to what services to offer. So the very first thing we did when we notified the groups is we sent out a survey and asked them, and tried to do a capacity assessment to understand what were their organizational needs, what did they need in terms of grappling with science or getting up to speed on the state of the science in their disease. And so we are shaping these things based on their responses. And so we're doing a bunch of things. We're going to be holding 3 in-person convenings over the 2 years, bringing the whole cohort together so that the 30 groups are really learning together as a network of networks. We're holding monthly webinars on different topics that they are selecting that they would like help in, and community calls where they can share learnings on a monthly or bi-monthly basis about what's happening in their disease area. We're setting up a mentorship program, a partnership with Faster Cures, to help to pair them with more established groups like the Michael J. Fox Foundation, groups like the Cystic Fibrosis Foundation, so that they can learn from these organizations that have come before them that have made a lot of strides in their disease area. Conference planning, just helping them with all taking away the logistics. So they don't have to do that. So they can just really focus on the partnerships and building partnerships with researchers and not have to worry about the logistics of conference planning. Science advising, Tracy, who you saw in the video, whose daughter had the 40,000 seizures, has just come on board and joined our team to actually lead the science advising program. So we're gonna be looking to match the groups. These groups don't need to hire a full-time CSO. That's just not practical for groups of this size. But what if they could have a part-time CSO, a fractional CSO, someone with scientific expertise that's relevant to their disease area, maybe not a researcher specifically in that disease area, but with relevant expertise, who can advise them, help them get up to speed on the state of the science, point them to the articles that are most relevant to them, et cetera. And just digital strategy, helping them, most of these groups don't have a CRM. They don't have, some of them need help with websites, everything, soup to nuts sort of digital strategy to help them build and connect these networks. And then a couple of other things were one of the tricky things in this, we think, is helping patients to, how can we help patients that don't have a scientific background understand the scientific literature and partner with researchers? What kinds of tools would be helpful for them? And so we're starting basically a sort of investigation into this whole space. And what we're looking to is to gather and integrate relevant data like publications, clinical trials, researchers from multiple, and lists of researchers from multiple sources, and then distill key data sets and present them in patient-friendly fashions. And we don't quite know how we're gonna do this yet, but this is sort of a new project. So this is just to give you a sense of the range of the 30 groups that we have brought on board. This shows the number of papers and the number of researchers in the space. So for Tango 2, it's 18. And for Project 8P, it's whatever that number is, 24,000. So there's a huge range in how much research has actually been done. And you could argue in a way for the groups where there are fewer research papers and less research, it might be easier for them to get up to speed. But for the groups where there's actually a fair amount of research, what should they read? How do you distill that? How do you help them understand what might be, where the gaps might be? And so this is just an example. Oh, I seem to be missing some of the images, unfortunately, they didn't come through. But one of the things we're looking at is CZI has an in-house tool that we've built called Meta. Meta uses machine learning to map biomedical research in real time. And it allows researchers, it was a tool that was built for researchers. It allows researchers to create customized feeds so they can follow emerging trends and scientific concepts or authors over time. And so they get these emails every day saying there's a new paper in this that relates to this concept you're interested in, right? So we're looking at how we might be able to use the underlying knowledge graph of Meta to assist patient organizations to identify researchers and scientific literature that's related to their disease area. And we're working to develop a proof of concept prototype for using data visualization then to highlight those key researchers and collaborations. And the data here is for chordoma. We've been partnering with different rare disease groups to get them to help us, what would be helpful? Are we identifying the right researchers? Which are the researchers you work with? How can we think about how to make this data useful? And some of the images are not there yet. They got left out of the slide. But so one of the images is just a map showing where are the researchers geographically located on the map. And another one is actually looking at connections, which researchers seem to be more collaborative than others, because they're actually already working in collaboration with other researchers. So if you wanted to prioritize who do you call first, you might want to reach out to the researchers who are actually already engaging in collaborations. So those are just some examples of some of the other things that we're, there's the map. Of the other things that we're looking at. So this is a list of top researchers, a list of different clinical trials. Can we start to integrate some of these different resources and in ways that become useful for research communities? And then finally, I just want to say that we recognize that what we're doing with the cohort is part of a broader ecosystem. And so part of our strategy is also to really help build the broader rare disease field. There are some wonderful umbrella organizations, NORD, Global Genes, Every Life Foundation, Eurordis, Genetic Alliance. We are exploring partnerships with all of them on like how can we both leverage the incredible resources they've created for this community already, how can we help support them to further the amazing work and support they're doing for the community as a whole. And also, you know, pattern.org, the Rare Genomes Project here at the Broad Institute, these are two initiatives we've actually funded and are working in partnership with. So those are just, so yes, so basically the idea is we recognize we have to actually be a partner in helping to build the field as a whole. And then this is really just an acknowledgement to the Rare as One team. We have an incredible team at CZI working on this, and this has all happened in 2 years. It's been kind of amazing. It started out as me, and my first hire was Ann Claiborne, who started in March of just 2 years ago. And in the last 2 years, we've had these incredible people come on, and usually coming to me and asking me to work on this program, because I think so many people at CZI are really inspired and excited about working with the rare disease community. And I just want to say that 2/3 of the people on this team have a direct connection. Either they're patients themselves or they have loved ones with rare diseases. And I think that's really important. And that's really important to me as I build out the team, that we're building it with people who really understand what it's like to actually live with rare disease and experience this. And so with that, I'll stop there. Thank you.
Okay, you have not had the chance to ask any questions as yet, but this is your chance. This is the time, so I'll ask all of our speakers to come up and join me here so we can have a little discussion, chat about everything you heard today. And I think Amanda will put up for those who are shy, it's possible to text us questions that you'd like to discuss. There's also some mic runners around the room who would be happy to come to you with a mic to ask a question. Thank you. So maybe I wanted to open it up. Thank you. First of all, it's been an incredible afternoon and a great treat to hear your stories. Thank you for your time today. I wanted to maybe start with, we state the problems in this space and the challenges that, of course, are many and remain 7,000 diseases, lots of patients around the world, all the organizational challenges. I'm inspired by a recent piece that you wrote, Tania, and about I think what people point to as a success story, and I think that would be the Cystic Fibrosis Foundation. So I was wondering whether you wanted to share what you have seen there, and you said yourself that maybe not all diseases, what fits one disease will not necessarily fit other diseases, but what can we learn from that success? And there are some broad connections to some of the successes in the CF space as well in terms of treatments, but how did the patients organize themselves to get to where we are now?
Yeah, so one of the interesting things about the CF story is that, the CF gene was one of the first genes to be sequenced or identified by Francis Collins way back, so in the 80s, right? But what was interesting is the Cystic Fibrosis Foundation had actually gotten established much earlier than that, I think in the 60s. And they had already really started organizing their community. And so then once genetic testing started for CF, they were already very well organized, and it helped because one of the first things they did was to sort of encourage everybody to get tested. And so what's amazing about the CF community is every single person in that community knows exactly which of the 1,800 or so I've lost, I don't know exactly how many there are now, mutations along the CF gene that can cause the disease. They know exactly which mutation that they have. And that was enormously powerful because they were able to build out a clinical trial network so that and that they could recruit patients like that because they had an incredible registry that listed everybody's genetic mutation, and all the patients knew it also themselves. And so this was enormously beneficial to industry when, and when Kalydeco was developed, that was only approved for 4% of CF patients. And so at first, but it opened the door to a whole bunch of a whole new strategy for treating the disease, and has led to now a situation where 90% of CF patients, it's looking like, are going to have really good treatments. I think nobody could argue that progress, which took a while to get started, but once it got started, it moved very quickly, and the patient community has been a huge reason why it was able to move as quickly as it could. So I think what's generalizable is that organizing communities matters, and has to be the foundation to everything that we're doing in this space, because without the patients, it's really impossible to make progress.
Yeah. I think it's a remarkable story, and I think exactly those are the learnings. Now, Patty and maybe also Eric and Sonia, in reaching out to organize communities, as you have all done in a remarkable way, I would imagine, as humans being humans, we're all different people, that people might have different agendas as you begin to try to reach out to them and organize them under an umbrella, whether that's beyond the diagnosis, for advocacy for all rare disease, or whether it's an umbrella in a particular rare disease, as in prion disease. So what were some of these challenges, and how did you meet them, or how can other communities learn from some of those experiences?
I think there have been a tremendous number of challenges. One thing we thought about really hard in the beginning, as we were thinking about the need to create a registry in prion disease, was the perils of ending up with a fractured registry landscape where there's more than one, and some people are duplicated, and you kind of never know who, and suddenly you don't know how these data sets relate or overlap. And so I think a very sort of time-consuming process in the beginning, but one that was ultimately really worth it, was getting together with the existing patient advocacy groups in prion disease and making the case that we all needed to approach this together once and hopefully do it right. And at the time, it felt like we were investing a huge amount of time in these very first steps. But I think that ended up being worth it. We definitely encountered a bit of, I don't want to say resistance, but just misunderstanding from people in the research community who we tried to involve early on, who maybe they had their own sort of spreadsheets of patients and carriers who they'd seen over the years, and saw this effort as somehow competitive with that. And we tried to emphasize the ways in which we were really hoping to create something researcher agnostic and much more general and useful, ultimately, hopefully, to them and to everyone. I think that message resonated more with some people than others. Mm-hmm. And it's just a communication challenge. Yeah.
I'd like to talk about it. I don't think it's a challenge that we've had, but it's been an unexpected occurrence with the art exhibit. I do a lot of research into my daughter's disease, just like every other parent. So I don't promote it. I don't, but I know an awful lot about Ehlers-Danlos syndrome, and in particular mast cells. So because of the exhibit, we will paint a child with a specific disease. Well, I have to research that. And eventually, or almost from the beginning, I become friends with the parents on Facebook. So I read everyone's post. And there was one mother whose son that we painted, Glenn, and he had KBG syndrome. And she had posted a picture of his legs. And she said, tomorrow my son's having his Achilles tendons cut. And, his legs are really tight, and something was ringing. Ehlers-Danlos syndrome is an incredibly multisystemic, complicated disorder. So it just didn't sit right with me. And I said to her, Annette, does your son have a sacral dimple? And she said, yes. And I said, does your son have this and that? And I said, I think your son has a tethered spinal cord. I would hold off on the, cutting his Achilles tendons. And Annette, being another brilliant, just like the rest of the moms in this space, went about researching and said, oh my God, she called me back about 2 hours later. She said, I think my son has a tethered spinal cord. It took her about 4 doctors, because oftentimes tethered spinal cord is hard to find on an MRI, and it needs to be clinically diagnosed. So they did get the diagnosis. They did the surgery. Her son had not walked in 10 years. He is now walking. He was having 100 seizures a day. He's down to about 6 seizures a day. His cognitive function has greatly improved, and it looks like it is a coexisting condition for many of the KBG kids. So almost as an aside of the work that I do, I've also found it in giant axonal neuropathy and Phelan-McDermott syndrome. And I keep reaching out to the moms that run these organizations. I'm like, wait a minute, and they're like, oh my God, I think you're right. First they think I'm crazy. And then they do the research and they said, oh my God, I think you're right. So now I've been polling these groups that we work with, because I have to learn this disease for the catalog, and I'm nosy. So I start researching it, and I got a D in biology. I'm gonna, flat out tell you I got a D in biology, but because of my kids, I can easily read a peer-reviewed medical journal article with no problems at all. And love is an incredible motivator. So what I've done is started mapping the connections that I'm making. I think that, I might not be a great scientist, but I am great at making connections. So I'm working with another researcher to kind of put together, if we can find the underlying cause with tethered spinal cord syndrome, we might be able to prevent it in the future, which means preventing seizures, preventing cognitive delays, or eliminating them entirely if we can get that cord detethered, or get it or prevent it. What among all of these diseases with known genetic mutations, what's causing this in all of them? And I think knowing that it's out there in so many different diseases is a key to finding out what is going on with that particular symptom. So, it's just been completely an unexpected journey for me not for my own children. I always knew that I was going to fight and I was going to study and I was going to learn. I'm actually taking edX courses in biology. But I didn't expect to be able to work in other rare diseases the way that I am now.
That's remarkable. So actually, this is an example of agendas aligning actually unexpectedly as well, not just,
exactly.
Which is wonderful.
Exactly.
And incredibly powerful, of course. So this could be for any one of you, but it's a formidable task to really try to help as many patients as possible with rare disease in each of your different missions. What are your thoughts about how do we actually scale this? How do we maybe... certainly for the CZI effort where you're really trying to be agnostic and help all patients, rare as one. But for any effort, really, how do we scale the effort? What are some of the secrets to making that happen?
So I think what we're trying to do is we're starting with 30 groups. We want to look we think things like you just described are going to happen, by the way. We think that these groups are already so excited to learn from one another. We're already finding that they're reaching out to each other and asking for advice on something, and then finding these commonalities and symptoms and things like that that they didn't know about, because they can't possibly know about all these other diseases. I mean, if the medical establishment and the research establishment doesn't know most of these diseases.
And it's only by chance that
why
I
would
know
they? If there
because
are
of the exhibit. Yeah.
Exactly. So I think it was David Feigenbaum who said once, there's 1,200 days of medical school and there are 7,000 rare diseases. Like, how would we possibly be trained in all these diseases? And so we're expecting like an incredible amount of learning to happen across. We're already seeing it happen. I think that's one of the most exciting things. But what we also hope is that we'll start to understand and learn from and with these groups as to what are the commonalities. I mean, we sort of have this idea that most of these groups, like building a strong patient, it's not, they're all going to have particular challenges. I mean, Sonia, you talked a little bit about that. There's always going to be differences. The state of the science is going to be in a different place from one group to the next. You could sort of see that just in the extent of research and publications on that slide, that huge curve, right? But what is needed, the infrastructure that's needed for success is actually kind of a little bit the same, or at least overlapping for so many of these diseases. You need a strong patient community. You need to find researchers who are incentivized and willing to work on your disease with you in partnership. You need, probably you're gonna need some kind of a clinical grade registry. And probably a biobank, cell lines, depends on exactly what your disease is, what biological specimens or whatever that you're gonna need. But the sort of the process of collecting those things, of being good stewards of those things, of learning how to manage a biobank, those are all sort of commonalities. And so if we can figure out those processes and that infrastructure layer and how to just make it easier for groups to sort of do those things. I think that's, and if there are roadmaps, if there's, you need these 5 things, if it becomes almost modular, we can help you do those 5 things, or here's who you need to reach out to do those 5 things. Here's how we can make those costs lower for everybody. And our hope is we learn with the 30 groups, and everything that we learn we'll make available to the broader rare disease community, whether it's tools that we develop and we'll put out into the world, whether it's training materials, et cetera.
Yeah, I'll just say that resonates a lot. I think as we've sort of thought about it from the perspective of how can we get private sector partners interested in a rare disease, because that is, at the end of the day, where a drug is going to be developed. It's been really exciting to see these sort of, you just used the word modular, like sort of modular targeted therapy like platforms coming online, right? As we look into like genetically targeted therapies, gene therapies, some of the tools. And it's not that there's going to be ever a one-size-fits-all technological solution, but some of these tools are going to be able to address a spectrum, right?
Yeah.
A spectrum of rare diseases. And I think the homework list that we were given, while it was in some senses tailored to the state of the art for prion disease, has a certain repurposable quality to it, where if we talk to other people who are trying to think about what would a company need to see from me, probably it's going to be some of those same elements, maybe some additional elements. And while our life certainly would have been easier if a company had just chosen to do all of those things and then develop the drug, I think one like silver lining of us doing all of that in the academic sector is that now it's sort of everyone's. And we can sort of like point to all these things and lay them out for other folks as examples of, I mean, we're far from done with all our homework, but here are some of the things you have to put on the platter, right?
That actually ties in with one of the questions that we received from the audience, and they were saying, to continue the discussion on alignment and scale, are there general lessons for how to convince biotech companies to get involved? Are they doing it because they see an economic argument in the long term? Are they doing it out of a sense of mission? Or being motivated by compelling personal journeys. I suppose it could be a mix of all of these things,
I
but
think
what
you need
in
all of
your
those things.
interactions with biotech partners, I guess, what has been the biggest lessons learned?
Yeah, I mean, I think it definitely takes the combination of like being able to speak really clearly to who is your patient population and how are you going to find them, and even if it's a small number of people, if you're really well networked, I think a lot of the risk for pharma is around, a tremendous amount of risk around having the right therapeutic target. And if you can de-risk that, then even a rare disease can become attractive. But I think there's also a lot of risk around time, just like thinking about the economics of their job. If you can get a trial launched faster or done faster in a way that's compelling to the patient community, the pharma deciders, and also like the regulatory people who are going to approve it, I think that it changes the whole economic picture to be able to do something swiftly. So I think that's one way setting the stage really helps.
So what about raising awareness, which, Patty, you have been a huge advocate for, and getting off the island, as you mentioned. I know you said that your explicit goal is to get us off the island and to really start to, connect with the mainland, I suppose. But what do you see as those next steps, like practically being? What would you like to see from anyone, parents, us, scientists here at Broad, doctors, anyone in the community?
I mean, I think that because we were so isolated for so long and there was so little interest in rare diseases, and I mean, now we're seeing much more growth as compared to even when I started 10 years ago. There's just been a huge amount of growth in this space. I think that it's just time now. We have got it in place, and we have got a lot of good models from different organizations that we can learn from and then move forward in that area. But what we need to do is now start thinking about how do we get more money for the NIH. The patient community is taxed. I can tell you as a mother, being a citizen scientist, being a caregiver, working in this space, most of these, like you said, most of these mothers are not getting paid to do this work. The financial stress is enormous. I think we need to not accept that as the business model for rare diseases. I think we have to move away from that and find ways like, how do we get interest with the NIH? And that's through global awareness. That's through having not just the rare disease community behind us, that's everyone behind us, just as they are behind cancer. I would like to see that kind of attention and that kind of, being able to be recognized, the zebra stripe, just like the pink ribbon is. That's what I want, and I think that's what we need. We can't keep relying on these mothers who are barely surviving to raise all of the money, to do all of the research, and get everything done, and still take care of their children. They're doing it, and they're superheroes in this space. They really are. You guys are remarkable. And like many of the people that I've met in this space, they're just remarkable. But I think now that we're in a bigger place, we're all in it. It's time to move forward with how we think about rare diseases and the responsibility of rare diseases. It's a global health crisis. When you have so many people that are suffering without treatments, that's a health crisis. And I think it's time that we move that forward and start talking more about that and less about stronger together. That's true. We are stronger together, but it's time to move on. We can't live like that anymore.
And from the vantage point of CZI, for example, how do you see policy and alignment of interests between philanthropy, government, institutions like ours, other medical institutions, and families and patient communities? How do you see that from your policy work and as you think about rare as one?
Yeah, so I agree. And one of the things I've struggled, I'll say it just point blank, one of the things I struggle with the program is the last thing we want to do is encourage the model where the more and more burden is being placed on the patients.
You won't be able to stop them.
But on the other hand, I actually think it's part of me wants to say, no, instead embrace the model, but make it possible for patients to do this in a way that makes sense, right?
Yes.
Pay them to do the work that they're doing, right? Like they have expertise that is incredibly valuable. I mean,
Yes.
we've just heard stories, like on this panel, about insights that patients had that led to new research questions that were actually the important questions to be asking that would have been missed, right? So I don't want to, but I struggle with, there's an ethical quandary, I think, in this around patient-driven research, which is this whole thing about patients driving research. Like, you know, and, you know, Tracy in the video, she doesn't say it in this video, she says in another video she did about her story, she's like, you know, a mom, I shouldn't have had to get a PhD to figure out what was wrong with my daughter. And she's right. But the fact that she did get a PhD changed her whole life and changed her daughter's life, and so, and is inspiring so many people. And so, like, yeah, absolutely, I agree. And so I think the way we get there and the way that I'm thinking about this is I want to help build and strengthen patient communities. They're not all going to drive research forward. They're not all going to change their careers and become researchers, and they're not all, certainly, and they're not all, but by strengthening those communities, that's how we can bring about the policy changes that we want to see. Yeah. So it's sort of like, I see what we're doing with Rare as One as part of a broader effort to strengthen the rare disease advocacy movement, that we need to build a movement. And it can, the storytelling that can come out of these stories, and by lifting up these stories and these incredible stories of what patients are willing to do and the motivation that they have, I think that does change hearts and minds, and that is what is needed.
Absolutely.
So I think it's some of all of those things, but I think I agree with you that that is the ultimate change that we all want to see.
It's wonderful, and actually acknowledged by one of the questions. I received a few of them now, about the incredible humanizing power of storytelling, paintings, movies, videos, and someone in the audience is saying, do we need more of that? And I think you were just saying how powerful that is, and how much more of that we need to build this movement, as you're saying. I don't think we have time to answer. Oh, we have a question, live question. Lovely. Yes.
It's working. I think it's working. Hi, hello, Raul Pina. I'm a clinical geneticist. I belong here. I work at Brigham, but I will try to talk not about Brigham and what I do. I'm trying to be an advocate for the Mexican community. So I have my practice in Mexico, and I will love to hear your opinion about this. I think we are not thinking that global, and this is a good example what is going on. Here, for example, this event is called Beyond the Diagnosis. In Mexico, I don't get a diagnosis there.
Yes.
Yeah. And I started not that many years ago, but for example, in 2011, I started genetics, my training. In that time, asking a researcher to get if they can do an exome, it was possible. Sometimes it's going to be Italy, sometimes it's going to be the US, but right now, asking for an exome is impossible. And many of the initiatives, one example is when I saw the initiative where it was like, wow, this is great we have some patients that are organized, but it's restricted to US institutions. And even the Center for Mendelian Genomics, the Undiagnosed Disease Network, and many of the things are restricted for US patients. So how you think this is going to change, or how we can help? Because this is creating a gap. So the majority of people that I see, I'm from the south of Mexico, is indigenous communities that they don't have Facebook, they don't have a computer, and when we ask an exome, we are asking about 1 year of salary to do an exome, or for some families, the Mennonite community I have there that I diagnosed a few patients recently, it's impossible. They need to work 5 or 10 years to get the money for a clinical exome. So we are getting money from projects, from donations to try to do, but the best that I could do as group, it's 50 cases or something, do just a small panel. So what do you think, what we will do for the future to decrease this gap that now is evident like this? In the US, we are thinking beyond the diagnosis, and in other parts, we're still thinking on the diagnosis.
No, it's definitely an excellent question, and the democratization of access do you have any thoughts, Tania, on the global reach?
It's an incredibly important thing. And so when we started this initiative, one of the first things we talked about was, should we start with diagnosis? Recognizing there are huge access problems, and diagnosis is a point of activation. Like, you can't become an advocate for your disease if you don't know what your disease is. So I completely agree with you. We felt a little overwhelmed starting there, to be honest with you. We were sort of like, wow, that is such a, and again, I kind of came back to saying the movement's not there yet. We don't have enough we haven't created a broad-based movement enough to get to, like even in this country, we don't even have access to like guaranteed health care.
Right.
So there are different problems, right? But I know that pales in comparison to what you're saying. But still, for some people, it's actually the same struggle, even in this country. So yeah, so I struggled a lot with where to start, and I think the access problems are huge. The other thing I struggle with is, like, who are the patient leaders who are emerging to lead these organizations? They are more privileged, and because, like, who can afford to quit their day jobs and do this work, right? So one of the things we did look at when we were designing this, we did have to start with 501c3s in the United States, and we wanted to make it global, but we just thought there was no way we could handle that many applications from around the world and even manage a cohort that was truly international for the first round. But ultimately, we aimed to make this a global initiative. But what we did look, and we asked a lot of questions about diversity and inclusion and how do you reach your patient community, and one of the things we really looked at in applications were groups that were actively reaching out internationally. So every one of the groups we funded, I'm pretty sure it's the case, has an international patient community and is actually at least engaging patients from around the world, and that was incredibly important to us as a starting point. It's not good enough.
That's not audible, but I can say, do you have any advice is the question. I mean, we grapple with this. I also see Heidi in the audience, and Heidi Rem is the leader of our Rare Genomes Project, as well as several other large sequencing efforts that are really going on around the world, bringing together scientists and investigators from everywhere. Clearly, there are some places where the resources are better in place for this to happen. That's clearly the reality. We can't really sugarcoat that. The aim, of course, is to reach all populations, represent all ethnicities and people from all over the world. It's a lofty goal, and it will not happen in one day. But I think at least the goodwill and intention is there. And many of us spend time on the phone in conference calls with colleagues in places like Australia and other parts of Latin America, South America, Europe, Asia, to try to coordinate efforts. The Broad has been a big part of that in several different areas. Clearly, we have a lot of work to do still, and there will be many challenges ahead. But I think we can maybe end on a note of hope that as we continue to work together as teams, scientists, patients, advocates, patient scientists in particular, we can really make progress in an area that is, of course, of much need. I wanted to thank all of our speakers today. It's been a remarkable afternoon, which hasn't ended yet. We're going to have a reception outside and, importantly, the unveiling of a portrait, which I can't wait to see. And so you'll also have a chance to engage with our speakers in a more informal setting, if you will. And I wanted to thank each and every one of you for coming today and making this day special, marking the Focus on Rare Disease Day at the Broad with your presence. Thank you very much.